Hi, welcome back to the second series of muscle physiology. We will be discussing molecular mechanism of contraction. We will focus on the roles of troponin, tropomyosin, and calcium in muscle contraction. So first look at tropomyosin. So tropomyosin molecules act as a regulatory protein that lies along the actin filament. In our resting muscle fiber, these tropomyosin molecules partially cover the myosin binding sites on each actin molecule. So this coverage prevents the myosin cross bridges from making contact with actin, and it will effectively block muscle contraction under normal resting condition. But there is more to the story. Each tropomyosin molecule is held in this blocking position by another protein called troponin. So troponin is a smaller globular protein that binds to both tropomyosin and actin, and this interaction stabilizes tropomyosin in its position. It will ensure that myosin binding sites remain inaccessible. Now here's a question for you to think about. Why are muscles not in a continuous state of contraction? After all the necessary components for contraction that are actin, myosin, and ATP, they are present in muscle fibers. So the answer lies in the function of troponin and tropomyosin, which prevents interaction between actin and myosin in resting condition. So this critical regulation ensures that contraction only occurs when it is actually needed. Now let's continue discussing the molecular mechanism of contraction with a focus on the critical role of calcium ions in regulating this process. So to initiate contraction, calcium ions bind to specific sites on troponin, not tropomyosin. Remember that. So this binding causes a conformation change in the troponin molecule, which in turn shifts the tropomyosin molecules away from their blocking position. So as a result, the myosin binding sites on actin are exposed. It will allow the cross-bridge cycle to begin. So this interaction is essential for muscle contraction. However, the system is not always active. When calcium is removed from troponin, it reverses the process. So tropomyosin moves back to its original blocking position on the actin filament, and it will prevent the interaction between actin and myosin. So this initially turns off the contractile activity. It will allow the muscle to relax. Now let's discuss the concept of excitation-contraction coupling, which is the fundamental process connecting the electrical signal in muscle cells to the physical process of contraction. Excitation-contraction coupling refers to the sequence of events by which an action potential, which is a brief electrical signal in the plasma membrane of a muscle fiber, triggers cross-bridge activity between actin and myosin, and it will result in muscle contraction. If we talk about how does this occur, step number 1 will be the process begins with an action potential which travels along the plasma membrane of the muscle fiber. This electrical signal acts as a trigger for the subsequent step. Step number 2, it will be there is a rapid increase in cytosolic calcium concentration. So this influx of calcium is crucial because it initiates the biochemical events required for contraction. Step number 3 will be then calcium ions then bind to troponin, a regulatory protein, which is this binding cause a conformation change in troponin, which in turn shifts tropomyosin away from the myosin binding sites on actin filaments. With the myosin binding sites now exposed, the cross-bridge cycling process can begin where myosin heads attach to actin, and it will generate force and it will produce contraction. And the last step, it is important to note that source of the cytosolic calcium is the sarcoplasmic reticulum. So a specialized endoplasmic reticulum in muscle cell is present, which is also called as sarcoplasmic reticulum. The sarcoplasmic reticulum stores calcium ions and release them into the cytosol in response to the action potential. So this tightly regulated sequence ensures that muscle fibers contract only when necessary, and the coupling of electrical excitation to mechanical contraction is precise and efficient. That's why we call it excitation-contraction coupling. Now let's talk about the mechanics of single fiber contraction, which highlights the relationship between muscle force and movement. Firstly, we need to define two key terms. Number 1 is muscle tension and number 2 is load. Muscle tension. So this is the force exerted by a contracting muscle on an object. You just can imagine pulling on a rope to lift a weight. So the force you apply is similar to muscle tension in a biological sense. And number 2 is load. So this is the force exerted back on the muscle by the object being moved, typically due to its weight, obviously. For example, if you are lifting a dumbbell, the weight of the dumbbell represents the load. So for a muscle fiber to successfully shorten and create movement, the amount of muscle tension generated by the fiber must exceed the opposing force of the load. If the load is heavier than the tension the muscle can generate, the muscle will not shorten, obviously, and no movement will occur. So this delicate balance should be present. Continuing with the mechanics of single fiber contraction, let's discuss the different types of muscle contraction and the specific characteristics. Number one is isometric contractions. So in an isometric contraction, the muscle generates tension without changing its length. So this occurs when the force produced by the muscle equals to the opposing load, so no movement is observed. Think of holding a heavy object in place without lifting or lowering it. Your muscles are working, but they are not shortening or lengthening. This is why isometric contractions are also referred to as maintaining a constant length. Number two is isotonic contractions. So in an isotonic contraction, the muscle shortens while maintaining a constant tension to overcome the load. This is the type of contraction that occurs when you lift a weight, where the muscle changes length but keeps generating consistent force to move the object. Number three is lengthening contraction, also known as eccentric contraction. So these occur when a muscle lengthens while under tension. So this happens, for instance, when you lower a heavy object or walk downhill and the force generated by the muscle is less than the load it's supporting. So these contractions are critical for controlled movement and resisting gravity. So the last one and fourth one is twitch contractions. Twitch is the simplest type of contraction. It represents the mechanical response of a single muscle fiber to a single action potential. While twitches are fundamental to understanding muscle behavior in a laboratory setting, actually real-life muscle activity involves more complex coordinated contractions. Let's just discuss about the types of skeletal muscle fibers and how they are classified based on two key characteristics. Number one is their maximal velocities of shortening, and number two is major pathway used to form ATP. So on the basis of maximal velocities of shortening, muscle fibers can be classified as fast fibers and slow fibers depending on how quickly they can shorten during contraction. And number two is factor is major pathway used to form ATP. So skeletal muscle fibers also differ in their primary metabolic pathway. They used to generate ATP, which is the energy currency of the cell. Now let's continue discussing the types of skeletal muscle fibers, focusing specifically on the fast and slow fibers. So the key difference between fast and slow fibers lies in their myosin isozyme composition. So these isozymes are specialized form of the myosin protein that determine how quickly the muscle fiber can split ATP. ATP splitting is crucial because it provides the energy needed for cross-bridge cycling, which powers muscle contraction. First one are fast fibers. Fibers that contain myosin with high ATPase activity are classified as fast fibers. These mean they can break down ATP at a much faster rate, allowing for a quicker rate of cross-bridge cycling. So these fibers are optimized for rapid and powerful movements such as sprinting or lifting heavy weights. However, they tend to fatigue more quickly. On the other hand, fibers with myosin isoforms that have lower ATPase activity are classified as slow fibers. So these fibers split ATP at a slower pace that results in a slower rate of cross-bridge cycling. While their contractions are less rapid, they are more energy efficient and they are resistant to fatigue, making them ideal for sustained activities like maintaining posture or long distance running. So continuing with the types of skeletal muscle fiber, let's now focus on the classification based on their metabolic properties. Number one, oxidative fibers. These muscle fibers are highly specialized for aerobic respiration. Oxidative fibers contain a large number of mitochondria. It enables them to efficiently perform oxidative phosphorylation, which generates ATP using oxygen. So this makes them ideal for sustained long duration activities like walking or maintaining posture, So these fibers are rich in protein called myoglobin, which binds oxygen and facilitates its transport within the muscle cell. Myoglobin acts as an oxygen reservoir, ensuring that oxidative fibers have a continuous supply of oxygen even during prolonged activity. The high myoglobin content gives oxidative fiber the characteristic dark red appearance, which is why they are also referred to as red muscle fiber. So this red color reflects their endurance capacity and the reliance on oxygen for energy production. Now let's talk about the second type of muscle fibers, glycolytic fibers. So glycolytic fibers are specialized for rapid high-intensity activities. Unlike oxidative fibers, glycolytic fibers have relatively few mitochondria. This means they rely less on oxidative phosphorylation and more on anaerobic metabolism for energy production. These fibers are packed with glycolytic enzymes and store large amount of glycogen. This setup allows them to produce ATP rapidly through glycolysis even in the absence of oxygen. However, this energy production is not as efficient or sustainable as aerobic respiration. Glycolytic fibers contain little or no myoglobin. which is the oxygen binding protein. As a result, they lack the dark red color seen in oxidative fibers and instead appear pale. This is why glycolytic fibers are often called as white muscle fibers. Now let's conclude our discussion on the types of skeletal muscle fibers by summarizing how they are categorized based on their key characteristics: myosin ATPase activity and oxidative or glycolytic capacity. So these two factors give rise to three distinct types of skeletal muscle fibers. Number one is low oxidative fibers, also called as type 1 fibers. So these fibers combine low myosin ATPase activity with high oxidative capacity, and they are designed for endurance and efficiency. It makes them ideal for prolonged activities like maintaining posture or long distance running. They are fatigue resistant due to their reliance on aerobic metabolism. Number two is fast oxidative fibers, which are also called as type 2A. These fibers have high myosin ATPase activity, which allows for rapid contraction along with high oxidative capacity. They serve as a hybrid. It offers both speed and endurance. So these fibers are suited for activities like moderate intensity running or swimming. And number three are fast glycolytic fibers, type 2B. So these fibers combine high myosin ATPase activity with high glycolytic capacity. It makes them specialized for short bursts of intense activities such as sprinting or weightlifting. And they rely on anaerobic metabolism and fatigue quickly due to the limited efficiency of glycolysis. Interestingly, there is a theoretically possible fourth type, slow glycolytic fibers, which will combine low myosin ATPase activity with high glycolytic capacity. However, these fibers are not found in nature, likely because such a combination will be inefficient for both speed and endurance. In addition to the biochemical differences we have discussed, skeletal muscle fiber also vary in their size and capacity to resist fatigue, which further make them different in their functional roles. Number one is the size difference. Glycolytic fibers generally have larger diameters compared to the oxidative fibers, so the larger size of glycolytic fibers allows them to produce greater force. It makes them more suited for short bursts of intense activity. However, this comes at the cost of endurance actually. So these muscle fibers also differ significantly in the resistance to fatigue, as for example, slow oxidative fibers are highly resistant to fatigue. The reliance on aerobic metabolism and their smaller diameter make them ideal for prolonged low-intensity activities like maintaining posture or marathon running. And number two are fast glycolytic fibers, on the other hand, fatigue rapidly. While they generate high power for short durations, their dependence on anaerobic metabolism leads to the rapid buildup of metabolic byproducts, and it limits their endurance actually. And number three is the fast oxidative fibers. They fall in between basically. They have an intermediate capacity to resist fatigue, and they offer a balance between power and endurance. And with this, that's all for today. Thank you so much for watching. Make sure to subscribe to Pill Talk for the latest coming videos.